Alright, this is Jahangir from MedPorium, and today we have part 4 of our lymphatic and immune system unit, in which we will discuss cellular and humoral immunity. Now let's discuss the mechanisms of humoral immunity. Now, as we discussed, humoral immunity is antibody mediated and relies on B cells producing antibodies. If you can recall from part 3, B cells will develop immunoglobulins Mu and delta. These immunoglobulins or antibodies bind to the antigen of a foreign pathogen and activate the B cell. Now, there are two different pathways for B cell activation. The first includes the T cell independent pathway, and the other includes the T cell dependent pathway. The main difference between the two pathways is the intervention of T cells. In T cell independent, T cells do not intervene. This is when the B cell receptors on a cell surface undergoes a process known as cross-linkage, in which two or more B cell receptors will attach to the pathogen's antigens, which are known as the T cell independent antigens, leading to the next process known as clonal selection. Remember, the attachment of multiple T cell independent antigens to the B cell receptor activates the start of B cell activation, and these T cell independent antigens are mostly found on the cell wall of a pathogen. Now let's discuss the second pathway, which is our T cell dependent pathway, in which T cells play a role in the B cell activation. As always, our B cell receptors will bind to the antigen and recognize that antigen. The B cell will then present the antigen fragment as an epitope on the MHC2 protein. A T cell known as a helper T cell will attach to the epitope and release cytokines such as interleukin, which will initiate clonal selection. Both of the two pathways have one thing in common, and they both activate clonal selection, which is just the proliferation of a clone of B cells. This clonal selection results in a differentiation of B cells to plasma cells, also known as effector cells, and memory cells, with the majority being plasma cells. Plasma cells produce a variety of immunoglobulins, while memory B cells can remember the antigen of a pathogen previously fought and initiate a rapid immune response. Now, what do these antibodies do? Well, they perform four main mechanisms. The first mechanism includes neutralization. This is when antibodies bind to the active region of an antigen of a foreign pathogen to prevent that antigen from attaching to healthy cells and harming them. The second mechanism includes complement fixation, in which antibodies will bind to the antigens of a pathogen, activating the complement system, which results in lysis due to the membrane attack complex discussed in part 2. The third mechanism includes antibody-dependent cellular cytotoxicity, or ADCC for short. This mechanism is activated when Fc receptors of leukocytes, such as natural killer cells, bind to the IgG antibodies, which are attached to the pathogen's antigens. The natural killer cells will then send cytotoxic signals towards the pathogen, ultimately killing them. The same action is seen in type 2 hypersensitivity, only against the cells of the body. The fourth mechanism antibodies perform includes precipitation, in which antibodies, primarily IgM, attach to the antigens from foreign pathogen to form the antigen-antibody complex, which prevents the pathogen from binding to the surface of healthy cells. This complex is big enough to be phagocytized. Now, what happens to our memory cells? Well, when a previously fought pathogen enters the body, the memory cell will initiate a more rapid immune response, giving rise to more effector B cells, which will secrete even more antibodies to fight off the pathogen. This is known as the secondary response. If we look at this chart, we can see a difference between primary and secondary response. Now, the primary response is the immune response to pathogen first encountered. In the primary response, there are more IgM antibodies than IgG antibodies, but both antibodies are being secreted at a low rate, and this is due to our B cells first encountering the pathogen never seen before, in which the number of plasma cells produced is not high enough to induce a rapid immune response. On the other hand, the secondary response, we can see that the IgG antibodies are being secreted at a very high rate in only 1 to 3 days. This is where our memory cells come into play, as they induce a rapid immune response, producing many plasma cells, which will then produce millions of antibodies. Now let's discuss cellular immunity. Cellular immunity, if you can recall, is cell-mediated and is a specific defense in which lymphocytes, primarily T cells, will attack and destroy pathogen as well as memorize the antigen of a specific pathogen. There are four different T cells. The first class T cells are known as cytotoxic T cells. Cytotoxic T cells contain a CD8 receptor and mostly kill infected cells and cancerous cells. The second class are known as helper T cells. These cells contain the CD4 receptor and aid in the production of B cells, cytotoxic cells, and the action of cytotoxic cells. Helper T cells are the army general of the T cells. The third class are known as regulatory T cells, or Tregs for short. These cells differentiate from helper T cells and release inhibitory cytokines such as interleukin 10 and transforming growth factor beta, which suppress self-reactive lymphocytes, which is important in preventing an autoimmune disorder. Regulatory T cells basically regulate the lymphocytes of cellular immunity, preventing them from causing too much inflammation. The fourth class are known as memory T cells, and as the name implies, these cells memorize the antigen of a previously fought pathogen to release a rapid immune response. Now let's discuss T cell activation. There are 3 steps. The first includes antigen recognition. This starts by our antigen-presenting cells displaying an antigen. Our helper or cytotoxic T cells will come in and attach to that antigen. Now, as we discussed, our helper T cell will have the CD4 receptor, which will bind to the MHC2 proteins found in the antigen-presenting cells, and these MHC2 proteins will carry a non-self antigen. Our cytotoxic T cells will have the CD8 receptor, which will bind to the MHC1 proteins found in every nucleated cell, besides red blood cells. And the MHC1 proteins convey an antigen that originates from the cytoplasm. The second step includes co-stimulation, in which the helper T cells or cytotoxic cells will express the CD28 receptor, which will bind to the protein B7 of the antigen-presenting cell. This process is important in verifying the attachment of a specific antigen and preventing the T cells from attacking the tissues of the body. The third step includes the secretion of cytokines such as interleukin 1 and 2, which are secreted by helper T cells or antigen-presenting cells. These interleukins stimulate the proliferation and differentiation of T cells, which is known as clonal selection, creating helper T cells and cytotoxic cells, which are effector cells, as well as some memory cells. Now, what are the actions of helper T cells and cytotoxic T cells in attacking a foreign pathogen? Well, helper T cells play an important role in humoral and cellular immunity. When they are activated, helper T cells secrete interleukins, and these interleukins can attract neutrophils, natural killer cells, and macrophages to the area of infection, as well as aid in T cell and B cell clonal selection. This is the reason why the human immunodeficiency virus, or HIV, is deadly, because HIV will attack the helper T cells and try to replicate its RNA to essentially make more copies of itself, which can lead to acquired immunodeficiency disease or AIDS. Cytotoxic T cells, on the other hand, are the only T lymphocytes that directly attack a cell. After recognizing the antigen of an infected cell, cytotoxic T cells will release a burst of chemicals aimed at killing the infected or cancerous cell. These chemicals include perforins and granzymes, which result in apoptosis of the target cell. The cytokine tumor necrosis factor has a similar effect on the target cell. Alright, and unfortunately that wraps up part 4 of our lymphatic and immune system unit, in which we had an introduction to cellular and humoral immunity. In this introduction, we discussed B cell activation, the actions of antibodies, the significance of memory cells, T cell activation, and the actions of helper T cells and cytotoxic T cells. And as always, a Pomodoro day make you a doctor someday. I'll see you next time.